Well, we're coming towards the end of our segment, and so I want to end with looking into the future. We are really at the precipice of a new era in molecular and personalized medicine. We're getting data from the human genome sequencing studies that are telling us that there are many mutations that drive breast cancer. In fact, we don't know even which mutations are driving cancer and which ones are just passenger bystander mutations. We're finding that there is a whole myriad of mutations that are infrequent, and we're also finding that the whole picture is cohesive. In other words, if you look at genomic data, gene expression data, or old-fashioned subtyping by ER, PR, HER2, or proteomic data, looking at the proteins and the ones that are kinases that are activated, that they all seem to be showing us the same thing. It's like the blind man and the elephant. We're looking at different aspects, but it all's coming together cohesively in one picture. But the challenge, of course, is translating that into some benefit for our patients. And there have been, at ESMO, some data presented on a very interesting approach that now many centers are doing, and that is personalizing medicine on the basis of gene sequence. Discovering targets that a patient had that you otherwise might not know that they have, and then referring them to clinical trials that are using these targeted agents. And I thought I would leave that open to the whole panel to discuss where their predictions are as to where molecular medicine is going to take us, and whether it actually is a conflict or complements healthcare reform in general, where we are going to have to make medicine much more cost-effective and accessible to our whole population.
Well, that's a wide open question. I think the future is bright. I wish we had every center, big center had like $50 or $100 million to set up such a center. And I think if we had a rich pipeline that we could then use that information and apply specific drugs to a specific tumor type and signaling pathway that's dominant, that would, really be very nice translation. Whether it works or not is a different issue, but at least it would be a very rational approach. And I hope that's where we get to. I think the other thing you commented on is, as we go forward, not only cost effectiveness, comparative effectiveness. So as opposed to shotgun medicine, at least there'll be some underpinnings for the decisions we make. And I think that can only lead, ultimately, I hope, to cost savings.
I think genomics is really the key to the future of oncology, and probably to every disease in medicine. What we started to do is starting to dabble in some genomics, and we've been sending some of our very difficult to treat patients, like the triple negatives, nothing that you give them, they have any response, they just march through. We actually started sending them to Foundation Medicine and getting back some reports which make some suggestions to us, druggable mutations, and also for referring to trials. And so we've tried that a couple of times, and it's, we have some patients who have come back with a list of mutations like this long. But I think you have to start to look at those things, and you eventually will hit on one which will be very helpful for you.
I think the point that was somewhat made at the ESMO meeting with the presentation are the large number of mutations that were found in the patients as part of their study, and that many of the mutations do not have a drug yet developed. And so we are perhaps way ahead of ourselves in the science in being able to discover the mutations rather than to treat them, and to think about an individual mutation and having a trial large enough to really get some meaningful information about specific drugs and mutations is going to take a lot longer than our ability to discover the mutations.
Right. The presentation at ESMO by Fabrice André was very interesting. They've now sequenced about 400, and they were able to successfully sequence most of them. They only found druggable mutations in a smaller fraction, and then they enrolled those on a trial and found the response rate was much higher than you'd predict. It was somewhere in the 20 to 30% rate. Was that right, Joyce? I think you had seen that presentation, and I think if that really holds out, that's at least a step in the right direction.
Yeah, we did a study, or TGen fully sequenced 14 metastatic breast cancer patients, and you do find some surprising things. It is difficult to figure out which are the driving mutations as opposed to ones that are there, but high-level amplicons are of interest, kind of analogous to the HER2 story, and patients do have, in the Fabrice André study, the FGFR1 was not uncommon, cyclin D1, and there are targeted agents, and one patient we found was a high-level BRAF amplicon, and she actually was able to participate in a trial of a MEK inhibitor, and in fact had a very nice response. Some of these cancers are so complex that single agents aren't going to do the trick, but I'm optimistic that we need to be able to perturb these cancers and then perhaps bring in some of the anti-PD-1. You know, if you can just, you know, get the tumor to stop producing even temporarily some of the, you know, very invasive and anti-immunogenic, cytokines, you know, for example, and then activate the immune system in the setting of some of that cell death with anti-PD-1, because it's, I'm not so optimistic that genomics by itself is going to be the answer. You know, I think that the biggest news at ASCO was the, anti-PD-1 to break tolerance against some of these cancers, and so that's where I think, but combining these strategies, I think, really is the hope.
Well, thank you all so much for an interesting discussion, and thank you for joining us on Onc Live on our inaugural breast cancer issue surrounding challenges in the management of breast cancer. Thank you for joining us, and thanks again to our panel.